You are tuned into the Dr. Tyna Show with Dr. Tyna Moore. For more, visit drtyna.com. On this episode of the Dr. Tyna Show, I am so excited because recently a study came out that I'm going to break down talking about how GLP-1s actually shift the intestinal gut microbiome into a more favorable, less obesogenic, less insulin resistant promoting microbiome. I have been talking about this for well over a year now in different podcast formats, on different people's shows, on my own show, inside my Ozempic Done Right University, which is my big course for clinicians, health professionals, and currently is still open to the public for a short amount of time. So if you have been on the fence about getting in there, I highly suggest you get in there now before the price goes up and it turns into a clinicians only program. But we have only had up until now conclusive data on mice and not in humans necessarily. There's been some speculative data, but nothing as solid as what I've dug up today. And I'm so excited because this is just mind-blowing. And this is why. If you're one of the folks, especially if you're a doctor, and you're still telling people that GLP-1s are a waste of time, that just you should exercise more and eat less, the intentional ignorance around these peptides, I'm honestly just done being nice about it. Like, I'm just done. Like, I'm just going to call people stupid from now on if it comes to that, because I can't tell you the number of doctors who still think that this is just some kind of quick Band-Aid. And granted, we've talked about this at length, especially on this show. I don't think it's being done right. And that's been my whole message the entire time and that's why I started talking about this in the first place, is I don't actually think it's being done right in many cases. But that doesn't mean that we vilify the peptide. This peptide is misunderstood. Its many mechanisms are not appreciated fully. And it doesn't just slow down your gastrointestinal motility, make you feel fuller longer, cause a decrease in appetite in the brain, and that's it. I literally, you guys, this is funny. I just had a vegan doctor come at me on Instagram. And so, of course, I had to go check out his content. And he was so wrong about so many things in so many ways. But I don't engage. I just, I saw it. I blocked him. I left it as is. But the funny part is that he's an obesity doctor. Like, that's actually his job. I think he might be an obesity surgeon. And he actually said that GLP-1s don't do anything to help you lose fat, that they just make you less hungry. Like, that's actually the mechanism of action as he understands it. And that's what he said. And I was like, really? Of all people, the ones using this peptide the most in clinical practice I'm guessing would be obesity doctors and endocrinologists. And he is still that short-sighted, even though he favors them, right? So this is where we're at. So I can see why you guys are frustrated, because whether you're a health professional or you're the general public, you're getting a lot of bad information. And then I can't even begin to tell you the amount of DMs I get on Instagram or emails I get from people of just some quackery, straight up. And sadly, most of it, and I'm so ashamed to even say this out loud, most of it's coming from chiropractors who have something to sell you and are trying to talk you out of the potentials of a GLP-1. And they've linked them to, they actually still think they're being made from lizard venom, which they were never made from lizard venom. The active component was identified in Gila lizard venom, and then it was synthetically created. But that's not what's in your Ozempic pen, guys. That's really just ridiculous. There's all kinds of conspiracy theories going around about what's in these pens, and it's just ridiculous. I had somebody actually come up to me this weekend at an event and say, "Do you really think that the you-know-what is inside the Ozempic, and that's how they're getting it into people?" And I was like, I can't engage the stupidity there. That's just blatant stupidity. But then on this other side is these really smart people who are still vilifying it and saying that the only thing it does is cause you to eat less, and that's it. And that's how you're losing weight. And they some really smart people, some people that you guys really dig, still think that that's the only way they work. And because they're saying this on their platforms. And if that's how they work, then the calories in, calories out model must be correct. I mean, it hurts my head. The intentional ignorance, it hurts my head. It's just like, show me you haven't done your homework without telling me, right? So I did an episode recently titled the 7 ways that GLP-1s improve metabolism that you and your doctor do not know. So go back and listen to that. I'll make sure it's in the show notes for you. And you will hear about how it actually heals your insulin sensitivity. It heals your metabolism and the mechanisms by which it does that. But a really, really big way it does this that I've been speculating for this past year plus, and that I'm so excited to share with you today about, is this paper. Correlation between, and note the word correlation, so at least they're being honest from the get-go. Correlation between intestinal flora and GLP-1 receptor agonist dulaglutide in type 2 diabetes mellitus treatment, a preliminary longitudinal study. So we're going to break this down a little bit, and I'm going to give it to you in human speak so that you too can share this out with your friends and family and help us end the ignorance around GLP-1 agonists that people really, really want to seem to hang on to. Nothing gets me going more than a practitioner who doesn't have a license to prescribe a thing, be it a peptide or a hormone, vilifying said peptide or hormone, and then turning around and trying to sell you a supplement. That's my favorite. And that's what we're seeing so much of lately. And don't get me wrong, I love my chiropractor brothers and sisters. Like, I love being a chiropractor. I'm not at all dissing the entire profession, but that's who I'm seeing do this the most. And just so you guys know, I have a whole video on my website called How to Find a Good Doc, where I break down for you the concept of fake naturopathic doctors, which licenses can do what. But we've got a whole lot of folks online right now who actually are not clinicians. They are PhDs, and they have doctor in front of their name because they are doctors, but they're not clinicians. And now that doesn't mean that some of them may not have some clinical training, but 99.9% of them do not. They're scientists, but yet they talk about clinical ideas and tell you clinical protocols, but they've never seen a patient or done any type of patient care. They haven't been trained in pathophysiology. They haven't been trained in physical diagnosis. They haven't been trained in differential diagnosis, and probably haven't been trained in lab diagnosis either. So, but they're called doctor and they have huge followings, and you guys think they're medical doctors. And then we also have a whole bunch of doctors who are doctors, and maybe a chiropractor, for instance, but they have no license to prescribe. Chiropractors can't prescribe anything. So that doesn't mean they don't understand peptides, because I know some rad chiropractors who really, really, really do understand peptides very well. But for the most part, if you can't use something, or you don't have someone in your clinic using it, and you don't understand how it works, and you don't understand the potency of it and the efficacy of it, it's really easy to vilify it and then turn around and say, well, buy my berberine instead. So, there is no Nature's Ozempic, and it doesn't exist. We might have something that helps promote the L cells to do their thing, but that's nowhere near what a GLP-1 agonist peptide will do. So don't get them confused, and quit letting the Kardashians and everyone else try to sell you GLP-1 in a bottle in supplement form, because it's not it. It's like, that would be like somebody trying to sell you an herb that does the same thing as insulin. It doesn't exist. And don't confuse berberine for metformin. Those two actually do a lot of the same things, but metformin is not a peptide or hormone. So anyway, I'm getting off topic. Let's talk about this study. So the highlights of this were that long-term use of dulaglutide can alter the intestinal flora in patients with type 2 diabetes. This is important because one of the things I've been saying about these peptides is that the long-term use is where we get the healing. So I'm not so concerned about people being on them for a long time, because if done properly, which is what I talk about inside my course, the long-term use could lead to more healing and more improvement and a myriad of things. Long-term use of dulaglutide may alter metabolic pathways in people with type 2 diabetes. Indicators of type 2 diabetes were correlated with the abundance of certain intestinal flora. So basically, there are bugs in our gut, and of these bugs, some of them. Now, I want to give a disclaimer here. Pounding probiotics is never the answer, in my opinion. I get asked this a lot. What about the Akkermansia probiotics? What about the probiotics being sold as GLP-1 promoters? I'm not a fan of pounding probiotics. I am a fan of altering the microbiome with food and with health, and sometimes using single strains to get us where we need to go for a short amount of time. But that's a whole other discussion. Pounding probiotics is not the solution, and taking them for long periods of time, I should say. Pounding probiotics for long periods of time and expecting something good to happen, because many of the organisms that I may or may not mention as I go through this quickly, are great up to a point and pathologic beyond that. Or it depends on the health of your gut. If your mucus lining is degraded and those bugs are now touching the internal cells of your gut lining, or you have leaky gut and they're getting through, they can become pathogenic. So it depends on where they are, and it depends on actually what part of the intestines they're in. It depends on how many there are. It depends on even differences in those same lineages of microbiome, or microbiota. So I can't really say that, this particular strain has always been shown to be good when it comes to type 2 diabetes and bad when it comes to type 2 diabetes, because again, it just depends on where it is, how much of it there is, what form it's in, and what part of what's the overall health of your gut. So what part of your gut lining is it touching or getting to or getting through? Hopefully that makes sense. That means we don't want to pound them, because it's not more is better always. Too much of a good thing could be too much of a good thing. So this is interesting. I'm just going to kind of read off some highlights here. GLP-1 agonists are presently used as first-line drugs for the clinical treatment of type 2 diabetes. It can regulate blood glucose by stimulating insulin secretion and lowering glucagon levels. I've taught you guys that. They used a special genetic way of sequencing to detect structural changes in the composition of the intestinal flora of newly diagnosed type 2 diabetics. Newly diagnosed, this is important, after 1 and 48 weeks of dulaglutide administration. Our research found no significant changes in the intestinal flora after 1 week in subjects with newly diagnosed type 2. Nevertheless, after 48 weeks of dulaglutide administration, the composition of the intestinal flora changed significantly, with a significant reduction in the abundance of intestinal flora. This is good, you guys. We don't want too much. I think what happens is people end up in this sort of SIBO, small intestinal bacterial overgrowth. They get a ton of flora. We don't always want too much. We want diversity, not abundance. Furthermore, we found that fasting glucose levels, fasting peptide levels, hemoglobin A1C levels, and BMI are also closely associated with intestinal flora. This reveals that intestinal flora may be one of the mechanisms by which dulaglutide treats type 2 diabetes. Now, dulaglutide is like a third generation GLP-1 agonist, and it's right before semaglutide came out, basically. So it's newer, but it's not as old as some of the OGs, but it's not as new as the semaglutide, and definitely not as new as tirzepatide. So some interesting parts of this. The number of genes present in the intestinal flora is 100 times more than that of the presently known human genome. It's also known as the second set of genetic code of the human body. Long-term studies have reported that the imbalance of normal intestinal flora can cause acute and chronic diseases in different systems of the body. We've known this. The imbalance is considered an intermediate factor in diseases associated with various systems. Therefore, it's crucial to maintain a good symbiotic relationship between the human body and the intestinal flora. Human intestinal flora can affect the abundance of good bacteria, such as those that produce short-chain fatty acids and regulate the concentration of metabolites and short-chain fatty acids in the body, thus affecting glucose metabolism. Basically, the bugs in your gut definitely have an impact on your metabolic health. The intestinal flora regulates the levels of human bile acid and endotoxin. This affects the level of intestinal hormone release, which is what GLP-1 is endogenously, it's an intestinal hormone, by influencing intestinal permeability and inflammatory responses, ultimately affecting insulin resistance. So this is one of the main ways that I suspected that GLP-1s work, is that they shift the intestinal flora to a more favorable flora, a less pathogenic flora, and thusly help improve metabolic health and insulin resistance. And this study shows a strong correlation with that. And lastly, they can slow gastric emptying speed, which is what we all know them to do, right? That's what everyone's saying. Oh, that's how they work. That's why they keep you fuller longer, and therefore you lose weight. But that is not all of it. That ain't it, you guys. In vivo experiments showed that gut microorganisms can alleviate diet-induced hypothalamic inflammation and improve leptin sensitivity via a GLP-1 receptor-dependent mechanism, ultimately affecting human metabolic function. That's exciting. Another study confirmed that GLP-1 receptor agonists can affect the composition and structure of the intestinal flora in mice, which has been implicated in the gut-brain axis. So your gut microbiome, its relationship with GLP-1 can have an impact on the HPA axis and the gut-brain axis. This is one of the big ways I figured this was working, because so many people have reported back to me, including patients, myself, my family, that their irritable bowel disease, that their inflammatory bowel disease, that all of that has improved since they've been on the GLP-1. But again, there's a sweet spot. And this is why I'm such a promoter of individualized dosing, is because given too much, and you really could push this over based on what this study's showing us. Nevertheless, no relevant study has investigated how the gut flora changes in vivo in humans when GLP-1 receptor agonist is used to treat type 2 diabetes, and whether altered gut microbes are associated with blood glucose regulation. So what they did is they looked at dulaglutide, which is a once-weekly GLP-1 agonist, and they tested them before, they tested them 1 week after starting their gut flora, and they looked at stool samples, they looked at several blood markers, and then they looked at the genetic profiling of the intestinal microbiota. And then they tested that 48 weeks later. So 48 weeks after initially starting, and these were all in new type 2 diabetics. And what they found was that fasting blood glucose levels statistically decreased gradually with the administration of the dulaglutide. BMI significantly decreased gradually with the administration of dulaglutide. Hemoglobin A1C, which is a 3-month marker of how sugared up your red blood cells are, statistically decreased gradually with the administration of dulaglutide. Fasting C-peptide level statistically increased gradually with the administration of dulaglutide. That's a good thing that it went up. And basically, overall, tons of different microbiota shifted. And the way that it shifted was the bugs that promote obesity, type 2 diabetes, and overall metabolic dysfunction, the abundance of those went down. And the abundance of the ones that promoted good insulin signaling and insulin sensitivity and good metabolic health either went up or stayed the same. And overall, the diversity increased. And it's important to understand why diversity matters. It's because we don't just want a whole bunch of one kind of bug. We want a very, very diverse gut microbiome. And one of the ways that we know that patients are falling victim to poor health due to their gut health is when we test their gut and we see that the diversity is very low. I have a friend who runs a high-level testing company, and he told me in 2022, I think it was, that the diversity of people's gut microbiome had significantly bottomed out over the few years prior to that. So since the beginning of lockdowns. And why is this? Well, people were locked at home and they were probably eating a lot of the same foods over and over. The diversity of our diet leads to the diversity of our gut microbiome. And people who really love to eat ultra refined carbohydrates and, just kind of the same thing every day are going to have a very low diversity in their gut, because meats and proteins and carbohydrates and plant sources of food, those are all going to increase diversity. So bottom line is we want a lot of diversity. And the other reason he probably saw them fall was because people were locked up and they were isolating and staying to themselves. We share our microbiomes, whether we believe it or not. We share our microbiomes, and thusly we share our diseases, even our chronic diseases that are not considered to be contagious, like heart disease and Crohn's disease, all kinds of disease processes. If people are genetically susceptible and they're hanging out with people who have said diseases, then they will share those through exposure and living in the same household. And, it just goes on, but none of that to scare you. It's just to say we actually want to be around a lot of people, diverse array of people. I would say healthy people. That's why I always say surround yourself with healthy people, because you will become the 5 people you surround yourself with the most. And it's not just because you adapt their habits and their lifestyle. It's because you actually share their microbiome. And when people were not hanging out with anybody else, they were not having a very diverse microbiome, and they were probably eating a lot of the same foods over and over again. Okay, so this study was small. I will tell you that. It started with 337 patients, and then they got down to 41 subjects that were enrolled in the study. So it's a small study. And again, the title says correlative. It's not causative, but it's very interesting, and it's pretty close to causative because they track things pretty closely. But they basically decided that after 48 weeks of treating type 2 diabetes with dulaglutide, there was a noticeable decrease in the abundance of the gut microorganisms in subjects compared to that before treatment, while the diversity of the gut microorganisms remained relatively stable and or increased a little. So to conclude, basically they looked at an alpha diversity and a beta diversity, and it was the same, if not more. In the beta diversity, it was significantly different. So that's great. That's great news. That's all very cool. Let's see, what are some of the high points of the discussion? There was a study done by Grassett et al., and they reference this in this study. I'll put the link to this study. This was in Cell, you guys. So Cell Press is a very prestigious publisher of studies and well-respected. And this is an article out of iScience. iScience is a division of Cell Press. It's a newer open access way to get studies. So I think that that's very cool. Grassett et al. Demonstrated that specific intestinal flora dysbiosis in type 2 diabetes in mice occurs via a gut-brain axis mechanism, which results in GLP-1 resistance. So this goes back to this conversation I've been trying to have about potentially a functional GLP-1 deficiency. And I get a lot of obesity docs and endocrinologists come at me and tell me I don't know what I'm talking about because I don't have the right letters behind my name, because I'm a naturopathic doctor and I'm a chiropractor, and they think I don't know anything. But I'm telling you, when physiology is off, and the pathophysiology of an individual is roaring, you can end up with functional deficiencies in certain signaling peptide hormones, directly in hormones, and then you can also end up with resistance. So the fact that GLP-1 resistance has been shown in mice, it's not too far off to speculate that that could be happening in humans as well. So you guys can check that out here. Moreover, the relative abundance of lactobacillus in the ilia of type 2 diabetes mice decreased significantly, whereas that of several others that were pathogenic increased significantly, with a strong negative correlation between the relative abundance of certain bacteria, Bacteroides, etc., and the concentration of GLP-1 receptors in the ileum. It's an inverse relationship. So the more of these pathogenic bugs, the lower the GLP-1 secretion naturally, endogenously. So thusly, a functional deficiency of GLP-1 in the body. Boom. Intestinal flora can play a role in controlling energy homeostasis in dietary fiber fermentation, thereby producing short-chain fatty acids. This is where something like sodium butyrate can come in handy when folks have misconstrued thinking that I said that sodium butyrate was nature's Ozempic. It's not. All I'm trying to share is that butyrate is a short-chain fatty acid, and it helps in the production of natural GLP-1 production. And so when intestinal flora is screwed up and the intestinal microbiota is in a more pathogenic lean, then we're going to end up losing energy homeostasis, and we're not going to have the functional, proper breakdown of our carbohydrates, our fibers, our starches, etc. And we won't get enough or as much short-chain fatty acid secretion as we need, because it's the bugs that make that. Intestinal flora in the human body regulates GLP-1 expression and intestinal GLP-1 secretion levels. Moreover, GLP-1 receptor agonist regulates the composition and structure of intestinal flora. That's very cool. Intestinal flora and GLP-1 seem to have a positive feedback relationship. Furthermore, when they did the 16S RNA amplification sequencing, showed a significantly decreased abundance and diversity of intestinal flora in rats, with a significantly decreased abundance of obesity-associated flora and a significantly increased abundance of lean-associated flora. So we're getting a better balance, and it seems like the GLP-1s, at least in these mice and rats, are doing very cool things to help balance out the flora. They also looked at another study that recruited 52 diabetic subjects who were divided into efficacious and non-efficacious groups based on whether GLP-1 receptor agonist administration was efficacious or not after 12 weeks of intervention. This is really important that you guys hear what I'm about to say. For those of you who've used GLP-1 agonists and said they didn't work, why didn't they work? I went all the way up to the top dose and they still didn't work. This study was small, 52 diabetic subjects, but what they found was that the diversity and the abundance of certain flora in their gut determined whether the GLP-1 agonist worked or not. So you guys can check out that reference. It's in this main study that I'll share with you the link to, and you guys can look at it. But depending on your gastrointestinal flora going into GLP-1 use, even though it is shifting your flora over time, if you're not doing all the things, you may or you may not have, any kind of efficacy with it. It may not work for you. And this is why I say we can't just rely on cranking up monotherapy of GLP-1 when there's so many other things that need to be dealt with. I talk all about gut health and the other peptide signaling hormones in my Ozempic Done Right University. You guys can find a link to that. I'll even provide a coupon code for you in the show notes so you can get a nice discount on the course. Get in there and learn all these things. Even if you're the general public and you think you don't need to know it, you do. I get messages from people all the time saying, I don't need your big course. I'm not a clinician. Why would I need to know all that? You do need to know all that because most of the doctors I talk to don't understand this stuff. So you have to become empowered through education so that you can go in and be your own best patient advocate and you can start to make some of these changes on your own. You don't need a doctor for all of this. A lot of it is just the way that you live, the way that you eat, the way that you do things needs to be in alignment with a good, healthy microbiome so that these GLP-1 agonists work in your favor instead of doing nothing or even worse, making you feel terrible. We talk all about different dosing strategies in there. I talk all about cycling and rotating. I talk about hormones. I talk about gut health. I talk about strength training and exercise. I talk about insulin resistance and different ways to drive blood glucose into your cells to be used as fuel. I talk about mitochondrial health. All of these things are important. Plus, there's a ton of expert interviews in there so that you can hear it from different people, maybe said in different ways so that it resonates. It's so important that you guys get yourselves highly educated when you go on this journey because it's not just about getting a pen for Ozempic and cranking up the dose. There are multitudes of reasons why it might not work, and it's important that you hear about them because that might be affecting you. And we want to make sure that you are getting the full bang for your buck when you use them. And if you're doing all the other things, including all the other peptides that I would suggest using in addition to, if you're doing all of those things, you don't necessarily have to crank the dose up. And that's the beauty of it. So you guys can check that out in the show notes, grab the link, grab the coupon code, and get to work and get yourself educated. To exclude the interface of several factors in the experiment, including disease, medication, and diet, we recruited newly diagnosed patients with clinical type 2, uniformly performed physical examinations that provided the same type of diet, blood and BMI indices for the patient before week 1, and after 48 weeks of dulaglutide administration were determined, which confirmed the hypoglycemic and weight loss effects of dulaglutide. So not just correlation, pretty causative if you ask me. Simultaneously, we collected feces samples from patients to detect differences in intestinal flora. They did this RNA sequencing before, after 1 week, and at 48 weeks, or after 48 weeks of dulaglutide. So it was pretty comprehensive. Further, we administered dulaglutide for up to 48 weeks and observed a significant change in the alpha diversity index, the beta diversity index, the microbial abundance, and the microbial abundance in the intestines of the type 2 diabetes subjects, which that abundance decreased significantly. But remember, the diversity stayed the same and or went up, which is good. After 48 weeks of dulaglutide administration, intestinal flora was significantly different from that before and after 1 week of dulaglutide administration, confirming that long-term clinical injection of dulaglutide may alter the composition and structure of human intestinal flora. So that's where the correlative word comes in for this. Anyway, very cool. They go through and they show you all the different bugs they looked for. They tell you the general mechanism of action of each one of these bugs. And if you actually go through and read it further, it tells you when and where these bugs, again, are favorable, but also where they can become pathogenic. So very cool. They talk about Akkermansia, Bifidobacterium. Bifidobacterium and Akkermansia, I think of them as like keystone bugs in the gut. And in both cases, we had a favorable outcome in those with the use of the 48 weeks of dulaglutide. So that's pretty darn cool. And then in conclusion, the intestinal flora of the human body was not significantly altered during the short-term use of dulaglutide against type 2 diabetes. However, during its long-term use, the abundance of intestinal microorganisms decreased significantly. The diversity did not change because that was the two ways they looked at it. In one case it did, and in one case it went up a bit. And the overall composition and structure of the flora changed significantly compared to those before drug administration. Additionally, fasting blood glucose level, BMI, hemoglobin A1C concentration, and fasting C-peptide levels were closely related to changes in the abundance of certain bacteria. Nevertheless, the mechanism of action of GLP-1 in type 2 diabetes treatment through gut microbes needs further investigation. So that's it. That is the breakdown of the study. I will link it in the show notes as always, and you guys can check it out for yourself. Share it with your doctors, share it with the naysayers. This is really exciting. I think this is where we're going to see hopefully more research go, because our microbiome drives everything and it controls everything. And when it goes sideways, all bets are off. I know there are those of you watching right now who had some kind of gastrointestinal upset. Maybe you went on a trip somewhere where you got sick. Maybe you had some kind of terrible, stomach bug, like a virus or something at home, and you got very sick and you sort of purged your bowels and you were never the same since. And in some cases we waste away, and in other cases people put on a ton of weight. And I have heard this from so many patients over and over again tell me, I was fine until I went through this, and then everything was messed up and I gained a ton of weight out of nowhere. And this is where the whole eat less, move more argument just pisses me off, because it's so short-sighted. It's such an ignorant way to look at overall health. And it really tells me that people don't have any clinical experience. If they did, they would actually have some compassion. It tells me they don't understand endocrinology. It tells me they don't understand clinical care, because I've even heard people erroneously go on platforms and say that, 12 weeks of dietary intervention reverses PCOS. That's bullshit. It doesn't. Once some of these pathologies really start rolling, we will see changes. Of course, we need dietary intervention. That's always core, right? That's always first and foremost and core, but that's not all of it. There's so much more to actually healing a patient and changing the course of things when we've had disruptions in gut microbiome, when we've had disruptions in the HPA axis due to the gut microbiome, when we've had leaky gut induced, when we've got hormonal imbalances happening. That's not just from the foods they're eating, but because of epigenetic, their epigenetic footprint that they were given by their parents, that they literally cannot change. Their genes were marked. We can impact the way our genes behave, and we can impact the way our gut bugs behave, and we can impact the way our hormones behave truly with the foods we eat. But that's not the entire story. And this is where I say, let's use the tools we have appropriately to give people a leg up to get the help that they need so that they can actually live in abundance and good health. So with that, I will leave you. If you guys enjoyed this episode, I would love if you would rate, review, and subscribe. I'm sorry I'm a little stuffy today. I just went on a trip and my sinuses have been a little funny ever since. So that's my microbiome in my nose, isn't it? But thank you for listening to me sort of stumble my way through this with a small font. And I hope that it was interesting to you. Go back and listen to the other episode where I break down the 7 different ways that GLP-1s impact our metabolism favorably that I guarantee you and your doctor do not know. And listen to my whole series about GLP-1s. I appreciate your time, and I will see you next time. Thanks for listening to the Dr. Tyna Show. This is a Wellness Loud production produced by Drake Peterson and mixed by Mike Fry. Theme song is by John the Guilt. You can watch the full video version of this podcast inside the Spotify app or on YouTube. As always, you can email the podcast at podcast@drtyna.com. That's DRTYNA. And if you like this episode, please rate, review, and subscribe on your favorite podcast app. You can also find all of my offerings on my website at drtina.com. For more shows by my team, go to wellnessloud.com. See you next time, and thanks for listening. This podcast is for general informational purposes only. It does not constitute the practices of medicine, nursing, or other professional healthcare services, including the giving of medical advice. I am a doctor, but I am not your doctor. No doctor-patient relationship is formed. The use of this information and the materials linked to this podcast is at the user's own risk. The content on this podcast is intended not to be a substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions.